Hi everyone, welcome to my channel Pure Biology. Today I come with the continuing video of the Golgi complex, okay, and this is the part 2 of the Golgi complex. And before this video I have in that topic I have already explained about the structure and the actual function, and here the functions of Golgi complex, obviously this one, but here the synthesis of lipids and proteins, what is happening to the synthesis of the lipids and proteins, this is actually discussing here, okay. So before going to this topic, again I want to say thank you to all of you who have already subscribed my channel and support me a lot, okay. So let's continue. First of all, this is the function, that is the glycosylation of proteins. One function that is offered by the Golgi complex, that is the glycosylation of proteins, and another function is that the lipid and polysaccharide metabolism, okay. So in case of glycosylation, the N-linked oligosaccharide chains that are the proteins altered as the proteins, okay. So actually here this chain is processing in the Golgi complex, okay. The proteins that pass through the Golgi cisternae from ER, okay. This in the ER, this type of protein that is produced, and for processing this enters within the Golgi complex. And the modification of in Golgi apparatus that gives two broad classes of N-linked oligosaccharides, okay. The complex oligosaccharides and the high mannose oligosaccharides. So what is saying here? The modifications of the in Golgi apparatus that gives two broad classes of N-linked oligosaccharides. That means that N-linked oligosaccharides, this is one kind of protein that enter in the Golgi apparatus, and here it after maturation or after processing, the two kinds of protein form. One is oligosaccharides and another is high mannose oligosaccharides, okay. And this high mannose oligosaccharide, the short form is, I have written here the HMA. This can contain 2N-acetyl glucosamines and a variable number of galactose and sialic acid, okay. This HMA that contain more than the original, the 2N-acetyl glucosamines and variable number of galactose and sialic acid, okay, sialic acid residues. In some cases obviously they contain fucose, clear. The complex oligosaccharides, these are generated by both removal of exiting sugars and addition of new sugars, clear. That means here the sugar particle is added or sugar particle is removed. By this process, the complex oligosaccharides, these are formed, okay. And some proteins that are related to O-linked glycosylation, that is O-linked protein, obviously you can say, or you can say that N-linked oligosaccharides, like that O-linked oligosaccharides, okay. This glycosylation is also occurring in the cistern, and here this O-linked oligosaccharides generally very short chain, and these containing only 2 to 4 sugar residues, okay. So here actually O-linked sugars are added one at a time, and each sugar transfer is catalyzed by a different glycosyl transferase enzyme, okay. This is possible by enzyme, this is called the glycosyl transferase enzyme, and here this is very short chain, and here the 2 to 4 sugar residue as present. That's why the sugars are added one at a time, okay, and each sugar transfer, this is a process, this is catalyzed by the enzyme. Here only the sugars that are added one at a time, this is main important, one at a time, and this is catalyzed by the enzyme. Clear? This is the actual glycosylation process of proteins, very brief process. Okay, I have explained here very briefly. Next one is the lipid and the polysaccharide metabolism. What is happening here? Here the ceramide is one kind of lipid that is produced by the endoplasmic reticulum and is converted either in sphingomyelin or glycolipids in Golgi apparatus. Okay, so what is produced in ER? Ceramide, and that enters into Golgi apparatus and then convert it into either glycolipids or sphingomyelin. Clear? In plant cell, many oligosaccharides that are processed or synthesized in the Golgi apparatus. Example is hemicellulose. Okay, hemicellulose. So these are actual the process, very brief process of lipid and polysaccharide metabolism and the glycosylation of the proteins. And here another description is that the transport of the proteins through the cisternae. How the transport of the proteins through cisternae is possible? There is a two model. One is the cisternal maturation model and another is the vesicular transport model. Okay, in the cisternal maturation model, what is saying that each cisternae that matures as it migrates outwards through the stack, when it comes out from the stack for maturation, then what happened? Each cisterna that carries the protein or the lipid particles that moved backward to the compartment. Actually, I am checking the word that is component or compartment, so it is compartment. Okay. So what is saying here? Each cisterna that matures, how it matures? When it, for maturation, it comes out slowly from the stack. And what happened to the proteins that is carried by the cisterna? That moved backward to the compartment for processing. This is the cisternal maturation model, and by this process the transport of the protein is possible here. This model says that. And in the vesicular transport model, what is saying that cargo means, you know that the protein substance or lipid substance that is delivered to each cisterna in turn by vesicles that bud from one cisterna and fuse with the next in the sac. What is saying that one cisterna that carrying the protein or the lipid particle that is transported from that cisterna by bud form. Okay, bud form, and this is transported to the next cisterna or the next cisterna of another stack or next stack. What is saying that? I am repeating this line again, that here the any protein particles that is carried by the cisterna that bud from one cisterna and fuse with the next in the next stack. Clear? This comes out as the bud form from one cisterna and fuse with the other cisterna of the next stack. In this way, the transportation, I think not next stack easily, in the same stack. I think so, same stack, because the next cisterna is possible. Okay, that means here this is saying that the one cisterna that carrying the protein or lipid particle that buds off from that cisterna to the next cisterna of the same stack. Okay, I think that is explained here. Okay, and this is the vesicular transport model. So this is the function of the Golgi complex. Second part of the function of the Golgi complex.